My name is Bob Anderson. I'm going to tell you about a phase 1 clinical trial of a new drug that's potentially being developed for celiac disease, NexVax2. The study is called phase 1 study to determine safety, tolerability, bioactivity of NexVax2 in HLA DQ2 positive volunteers with celiac disease following a strict gluten-free diet. The study comes from Australia, and we're based at the Walter and Eliza Hall Institute and Immusan T, a company based in Boston. So I'll just briefly tell you about celiac disease. Celiac disease is a common condition which affects the intestine in about 1% of the population. And it's marked by damage, villous atrophy, crypthyperplasia in people when they eat gluten. The only treatment for the condition at the moment is a gluten-free diet. There's no alternative. But there are a number of problems and limitations associated with the gluten-free diet. There's often poor compliance because just as little as 50 milligrams of gluten, a hundredth of a slice of bread, is enough to damage the bowel. And in many patients, in fact probably about half, even at 5 years on a gluten-free diet, still have damage. Failure to heal the gut is associated with more osteoporosis and even a slight increase in cancers. So there are a number of limitations to gluten-free diet. Sometimes there are nutritional deficiencies, and it would be attractive if there was an alternative that was more convenient and more effective, but still very safe to treat celiac disease. So our work has led to the design of a drug using peptides. If you look at why celiac disease occurs, it only occurs when people eat gluten and only if they have the appropriate genes that encode HLA DQ2. The gluten is not well broken down in the small bowel in the human and can be absorbed in small amounts and modified by an enzyme called transglutaminase, which generates small reactive sequences recognized by T cells. We've been able to identify 3 key peptides from gluten that account for the majority of the T cell reaction to gluten and celiac disease in people who have HLA DQ2. So this is about 80% of the population with celiac disease. It accounts for a substantial proportion of the toxicity of gluten. Now, if you know what those key peptides are, you're in a position to consider developing various drugs, including vaccines. I'll stress again, though, that this drug was developed specifically for the 80% of patients who have HLA DQ2, but not other susceptibility HLA types in celiac disease. So we've developed NexVax2 as an equimolar mixture of 3 peptides that are between 15 and 16 amino acids. They derive from wheat and barley, and there are similar peptides found in barley as well. We've developed the drug as a mixture in saline. We've taken the drug through toxicology studies, through extensive studies for the solubility and stability, and we've shown proof of concept in a mouse model of the celiac T cell response. And so we reached the point where the drug was potentially ready for testing in humans. The drug, in the end, could end up being a very effective agent that desensitizes patients in the way allergy patients are to gluten. It could also be useful in diagnosis by allowing us to measure the T cell reaction in celiac disease, and in the longer term it may be useful as a true preventative vaccine, as well as guiding the path for other similar treatments and other immune diseases. So our aim in this study was to evaluate the safety and tolerability of weekly intradermal injections of NexVax2 and to establish the bioactivity of the drug, particularly with regard to the T cell recognition after injection. So the design of the study was to look at patients who were on a long-term strict gluten-free diet who possessed HLA DQ2, and they were randomized blindly to receive either placebo, saline injections, or 9, 30, 90 micrograms of NexVax2 weekly injections over 3 weeks. We took serial blood samples for safety tests, as well as to measure the T cell reaction in blood to the drug itself. And that was intended to allow us to measure whether the drug truly targeted the T cell population we predicted it would on our previous work in human subjects. The study lasted for three weeks with injections, and we observed all patients carefully. Overall, the drug was well tolerated and it was safe. However, we did note that there were gastrointestinal adverse events that were more common with the higher doses of the drug that we tested, 60 and 90 micrograms. 7 out of 19 subjects experienced nausea. 2 subjects were administered antiemetics, and in fact 2 subjects, in addition to that, vomited at either 2 or 5.5 hours after the initial dosing with the drug. And there was 1 further subject who received 90 micrograms who withdrew due to gastrointestinal symptoms graded severe. It appeared therefore that the drug was capable of inducing symptoms similar to that observed after oral gluten challenge, but only at the higher dose levels tested, 60 and 90 micrograms. Importantly though, we were able to show that in some patients, in 4 of the subjects, 1 who received 30 micrograms, 2 who received 60 micrograms, and 1 that received 90 micrograms out of these cohorts of 6 that received each dose level, generated T cells in their blood, typically 6 days after the initial dosing, but in some cases the T cells were there for longer periods. And so, in conclusion, this phase 1 study, which was intended to establish safety, tolerability and bioactivity, did indeed show that the drug was worthy of further clinical evaluation. Importantly though, intradermal injection of microgram amounts of this peptide-based drug, NexVax2, induced symptoms in some patients that closely resembled those seen after they would have ingested gluten. And on top of that, as we see in patients who've eaten gluten, 6 days after they had this injection of NexVax2, there was evidence of NexVax2 specific T cells that produce gamma interferon circulating in blood, supporting the notion that this drug does target the gluten-specific T cell population in individuals with celiac disease. This is a key step on the path towards the clinical development of this drug as a potential desensitizing agent with the potential goal of replacing gluten-free diet for the treatment of celiac disease.